The last speaker for this morning is Professor Oren Kobiler from the Gray Faculty of Medicine. He will tell us about recombination in herpes viruses.
Okay, so I will move back to viruses. Viruses, as you all know, are one of the major sources for developing the center for pandemics. And I'm going to talk about a virus that we don't think often as a causative agent for pandemics, although it has the potential, and we are talking about herpes viruses. Herpes viruses are usually what most people think about when they see blisters or cold sores on the lip. This is herpes simplex 1, the major cause of this lip infection. Most of the people in this room are infected with herpes simplex 1. Some of you also have herpes simplex 2. This is the second virus from the same family. They are very closely related. Again, infecting 60% and 30% of the population. Herpes simplex 1 is going down in infection numbers in the global environment. However, simplex 2 is going up. And we see that in the last few years. Most importantly, we think of herpes 1 as the upper lips infection, mainly around the oral cavities, and herpes 2 in the vaginal. But recent studies have identified that you can find a lot of herpes 1 in the genital herpes. So there is an anatomical breach of the barrier that we thought about as distinguishing between the two viruses. These viruses are very, very common and can cause a very, very severe disease in some people, especially in immunocompromised, in in infants, and for some unlucky individuals with completely healthy, otherwise healthy. They can also have what we call encephalitis. The virus enters the brain, and then it's a very, very deadly disease. With treatment, we get about 10% mortality, and without treatment, it's about 70% mortality. So this is a very important virus that can cause a very significant burden on of disease, and we have mainly one option of treatment. This is the acyclovir, the Zovirax, that you have as a cream to put on these cold sores, but it's also can be given as a intravenous drug, which is used for encephalitis and so on, and these are the most common drug. Unfortunately, we have some resistant to this drug, and then we have.
Can you remind close to the microphone?
I will.
The recording is not working.
Okay, so I'll try to stay near the microphone. Sorry about that. So, as I was saying, we have resistant to this drug. We are developing new antivirals in different combinations of antivirals, but acyclovir is still the most commonly used and drug, very, very successful in many cases, but I want to talk about a different aspect of this drug today. And to do that, I will go into the molecular level of these viruses and how, I will try to stay in place, sorry about that. I will try to get into the details of the molecular level of these viruses. So herpes simplex viruses are, this way, are large DNA viruses. They have a DNA genome, not like the corona. The genome is DNA, double-stranded DNA, huge virus in the realm of viruses. If you think about viruses, usually we are talking about something very, very small with small genome, about 10 kb, and these are 150, so they are much larger, have a lot of protein they encode for, and they have a very unique genome. So the genome is, as you can see here, has what we call the unique long region, the unique short region. Each region is, has repeats in the end, which is the terminal repeat. So you have the terminal repeat long, the internal repeat long, the terminal repeat short, and the internal repeat short. And these are all flanked by a region that we call the A sequence, which can switch the genome around. So you have 4 isomers of the genomes. So you get, and this will, I'm sorry I'm going to the details, but this will become important later for this talk. But this is the genome, and it's a complex genome. Again, most of the genes are in the unique long and unique short region, but some of them, or some of the most important ones, are in the repeat regions. The virus enters the cell, so this should be a cell, okay? And the virus enters on the membrane, then fused inside and moved to the nucleus, where it injects its genome into the nucleus, and the genome starts expressing the immediate early and late genes, which are all part of this replication cycle, and they are forming replication compartments within the genomes. And each replication compartment in the cell starts from a unique, or from one genome that entered the cell. And this is how this virus grows. The replication compartment has the entire replication machinery. I will try not to move again. But it has the entire replication machinery encoded by the virus, but it's not enough. And in the replication compartment, we also have many host proteins that are involved in the replication process and in the expression process. So you can see here that many genomes, many host proteins, and this is a list of host proteins in one study that was shown to be in the replication compartment, acting on the replicating genomes or connecting to the replicating genomes. And the most important group, the most important group for this talk is the DNA repair proteins that are found in this replication compartment. And why is that important? It's because these viruses, unlike RNA viruses that have lots of mutation that during replication, these viruses have a DNA genome, therefore the amount of point mutation they obtain is much lower than RNA viruses, and to compensate on that, they use a lot of recombination. Okay, so here is a study looking on 17 different isolates of Herpes simplex from different humans, and all of them are compared to one lab strain, and you can see that there are areas that are very, very similar to one of the genomes or other genomes, and it's suggesting that we have a lot of recombination that we can see between the strains that we isolate, and we can also see that in vitro, and this is what we do in the lab a lot of time. We took 2 strains and look for recombination, and this is what we are trying to study in our lab, what affects the viral recombination. And as I told you, we have different replication compartments in the same nucleus, and in order for recombination to take part, we actually need the interaction between the replication compartment. So here we labeled DNA from one strain and from another strain we made in the lab, one with green and one with red. And when they mix together, you can see a break that turns out to be yellow. And this is the site we think recombination happens. And we think that recombination requires both viral and host protein to be involved to allow this recombination. Again, this is how we think about this recombination process. So we decided we want to develop a new assay to study recombination and recombination rate. And this is based on fluorescent proteins. Previously, in our lab, in another lab, we used 2 colors recombination assay, which had one fluorescent protein in one area and the second fluorescent protein in another area. And then we looked for progeny viruses that came out with the 2 colors and called them recombinants. Now we added a third fluorophore in the same place as the one of the fluorophores in the second strain, and that allows us to get different more recombination type, so we can look at different recombination. The assay is very simple. We take the 2 viruses, we infect cells, we collect the viruses that come out of the cell, and we then replate them for individual plaques, and we see something like this. So now we took the parental ones are the red ones that you hardly can see here, and the one that are blue and green, which look here like very pale blue, if you can see them. And we can get a lot of different kind of colors of the plaques that are forming. These are actually 7 different plaques. So we have the parental ones, the red and the turquoise. We have the one that recombine, so classic homology mediated recombination. So now we have the blue alone, or the red and the green together. So this is classic recombination. homology mediated. And then we have the weird stuff that we also get. This is non-canonical recombination, things that have all three fluorescent protein, the blue and the red only, which should be in the same place. So this is kind of duplication, or the green only, which somehow got rid of both the red and the blue, suggesting a deletion of some kind or something like that. So the next thing we will go back here, to quantify this assay, we actually need to count all the plaques in all of the colors, and we decided that it's a labor-consuming, very labor-consuming process. So we used AI to try to get a much clearer or a much more faster way to count this plaque. So we, in the lab, developed an AI tool that can actually detect these different plaques and in good assess the color of each plaque as they come out. You can see that it can even distinguish between very closely related plaques. It can tell that there are two different ones, and he's giving quite robust results. Here you can see that how we did it. So we took 255 images, each of them about 100 to 300 plaques in each, and we trained the AI model on about 180. We then validated that it can read it, and then tested it on 23. And you can see that the quality of the labeling of the AI in assessing the amount of parental or homology-mediated recombinants is very, very good, very similar to what was counted by the students. The non-canonical are very low in number for each image. So one mistake make a lot of difference. So that's why the non-canonical accuracy is much lower. But as I will tell you in a moment, we still can see some differences in the non-canonical recombination as well. So we took a lot of images, a lot of experiments, and we clustered them here together to give you a sense of the frequency of each plaque that we get. And you can see that in most cases we get mostly the parental ones, about 35% of the population for each. 10% for each of the homology mediated, so about 20% of homology in this class, and about 1% from each of the non-canonical recombination events happens. The list of them is the yellow one that is losing the color. One of the colors, okay? So the next thing we wanted to see is that we can actually see differences. And the easiest way to do it is by looking at something that was already known, that homology-mediated recombination requires higher multiplicity of infection. Again, thinking about the virus that needs to meet in the same cell, in order to recombine. Therefore, when you increase the multiplicity of infection, MOI, you get more recombinants, and we usually do the experiment in 20 to get about 25% of the plaques recombining in homology. We can also see some dependence on of MRI on the non-canonical recombination, but this was not statistically significant because, again, this is very low numbers and very noisy, as you can see. Next, we decided to do some of the genes involved, host genes involved in DNA repair mechanisms have effect on recombination rate. So what you see here is, sorry, okay. What you see here is 3 different host protein involved in recombination, and you can see that they don't have a lot of effect on the virus that comes out, the titer of the virus, and they don't also have any significant effect on recombination rate, not the homologous one, and not the non-canonical events. So these have very minor effect, and we will talk about it in a little bit. This is a very important protein in recombination, known as, in a homologous recombination in the cell, known as MRE. MRE is involved, one of the key factors in homologous recombination in cells, and you can see that it has a small effect of increasing the non-canonical. So when we inhibit it with a factor, with an inhibitory factor known as mirin, it's a small molecule that can inhibit this MRE, we can see increase in recombination rate a little bit, but only in the non-homology. So if you stop the homology, you get more non-homology recombination. The other interesting protein, host protein that we found is called PCNA. PCNA, again, is a very important protein in many host replication machineries and repair machineries, known to be associated with the virus. Replication compartment is one of the most enriched proteins connecting to the virus replication. And you can see that when we inhibit it, when we inhibit it, we get a decrease in viral titer. And surprisingly, while we get a decrease in viral titer, we get an increase in recombination. And this was completely unexpected because usually we think about recombination needs to occur during the replication process. So this is really was a surprise to us that you can decrease replication and with that increase recombination. And then we ask ourselves, can other inhibitors of the virus can also increase recombination? And to our even more surprise, we found out that using a cyclovir, the drug that everybody uses, actually these are concentrations that decrease the virus yield by up to 2 logs. These are concentrations that are used in the clinics, not for encephalitis, but for other stuff. In this case, you can see an increase, about 30% increase in recombination rate of the virus, and a decrease in the non-homologous recombination or the non-canonical events. So you see a decrease. To see if it's unique to a cyclovir, we decided to look at another common inhibitor of the viral polymerase. It's called PAA, phosphonolacetic acid, and this again decrease viral titers, increase recombination, and again decrease the non-canonical recombination event quite significantly. Again, about 20 to 30% more recombination rate. So this is the polymerase inhibitors, and we thought, okay, we have now a new drug that is being developed, actually being approved for cases that are have tolerance to acyclovir. These are the helicase permease inhibitors. These are two new drugs approved in some countries already, and they also, in the concentration we tested, increase recombination and decrease the non-canonical recombination. So this again suggests to us that inhibition of the replication process or the protein of the virus involved in the replication process will increase the amount of recombination we get. And to check if it's true, again, by different mechanism, we looked at hydroxyurea. Hydroxyurea decreased the nucleotide in the cell, and therefore lesser DNA replication can take place. And again, it's decreasing the virus, increasing recombination to less degree than before, but still you can see that, and has no effect on the non-homologous recombination or the non-canonical recombination. Okay, then we decided to see if it's also true in a different cell type to make sure that it's actually occurring in two different cell types in the lab. So we tested different cell types, and we see the same thing for the homology-mediated recombination. Again, an increase. This is with acyclovir and with pertrelavir, the helicase permease inhibitor. Both of them, again, increase recombination. In this cell type, we don't see any effect on the non-canonical recombination. So again, the non-canonical is not as robustly significant as the canonical, which it's much easier to measure in our assay. Okay? So the next thing we wanted to ask, does it affect the viral replication? So we looked at DNA copy numbers. First, again, we saw the viral yield is decreased, but we looked at viral genome copy numbers, and you can see that the drugs, although inhibiting the replication complex, do not decrease the amount of DNA we get, suggesting that what we get is more genome copies per plaque-forming unit, suggesting that we get a lot of defective viruses instead of normal virus that can replicate. And to show that we don't inhibit the DNA replication, we also looked at recombination or the replication compartments. And you have to believe me because you can see anything here, but there are red dots in the middle of these nuclei, and with treatment and without treatment, they look the same. I guess you cannot see them at all, or I cannot see them from here, but you will have to believe me that they look the same, and this suggests to us that the replication compartment is still functioning in this concentration of the drug, and replication is still taking place, but we get more recombinants due to these drugs in these concentrations. So we wanted to see if it also affects recombination between herpes simplex 1 and 2. We know in the last few years there were some evidence in the clinics for recombination between the two viruses, and we wanted to see if this is indeed we can recapitulate it in the lab. So we took again dual color herpes simplex 1, you can see here, that has a red fluorophore and a green fluorophore, and herpes simplex 2 that doesn't have any fluorophore at all. So, and you can see here that these are the parentals, and when they recombine, you get one that is red or one that is green. And you can see this recombination. And when we look at it, when we look at it, we can see that recombination does increase with these two drugs, acyclovir and petrelvir. Again, a much lower number of recombinants that we can see. Recombination between the two viruses is less frequent than within the same virus because they are different in many ways, but we can still see an increase due to the drugs. So we tried to look at these differences, and what we have done here, and again, it's a little bit hard to read, but what we see here, we took all the plaques that came out with one color, we isolated them and asked what kind of mutation in their genomes we can detect. So we did a very simple, old-fashioned PCR, very simple but a lot of work, PCR study. So we took 20 genes of the virus spread evenly between the UL, or evenly between each other, but in the UL, the US, and in the repeat region, and looked to see in the plaques if we can see what comes from HSV1 and what comes from HSV2. So there is a size difference between HSV1 that is higher than the 100, and the HSV2 is at the 100 kilobase PCRs. And you can see here, for example, that almost all the UL region in this plaque came from HSV2, and the US region came from HSV1, okay? So this is the assay, and you can see that when we give the drugs, you can see weird stuff happening, like here in this one, that you have HSV1, then HSV2, then again HSV1, then again HSV2, and some deletion also. So putting the drugs increase recombination, not only in what we can visualize, but also in the amount of aberrations that happen per genome. And we can see it quantified here. Here the acyclovir is significant and the petrelvir is not significant, but this is definitely something that we can see. So what it suggests to us that we actually increase the rate of recombination due to these commonly used antiviral drugs. This is a new thing that we didn't know about. We should take much more caution when we treat, especially over-the-counter drugs that can result in both drug resistance and new variants that we don't know about. recombination between the two viruses that we don't know about it. We couldn't find anything really, really significant with the host protein. We think that this is due to some kind of redundancy between the host protein because there are so many of them that some of them can contribute in different ways. So we think that's why the reason that inhibition of one of them didn't show any significant difference, but the drugs that block or inhibit the replication process of the virus, they actually cause the virus to probably some replication stress, and to reveal this, to overcome this replication stress, it actually recombines with different strain in the same genome, and that's what increased the rate of recombination that we see. That is our ongoing hypothesis. And with that, I want to thank the people in the lab that did the work, especially Maya, sitting here, over there. I won't put a laser pointer on you, sorry. And other members of the lab that help her in this process, our longtime collaborator, Matthew Weitzman, and the founding agency, the Israel Science Foundation, that helped do this work, Yuval, that developed this AI that really helped us make this a much more robust assay that can be easily quantified, lots of reduction of labor. And with that, I want to say that we are 600 days waiting for them to come home. So